All right, welcome to WSQ number 21, Advances in Genetics. Today we're going to talk about what we've learned, excuse me, what we've learned in the last couple WSQs and apply that into this last section, talking about the ways that science has actually taken genetics and what we understand about organisms to grow and be able to use that technology for new applications. So we're going to learn about the 3 ways of producing organisms with desired traits, one of which is a very old idea and some that are not so old. And then we're going to talk about 2 applications or more of DNA technology in human genetics, so what we can see going forward. So let's jump into it. First thing I want to do is talk about the 3 different ways that we see, as we see this first objective, the 3 different ways that we can produce offspring or organisms with desired traits. The first of these 3 is something that's been around, honestly, for a very long time. It's called selective breeding. And what selective breeding is the selecting, okay, that's where the word selective comes from, the selecting of organisms with desired traits to mate in hopes to produce offspring with those same desired traits. Okay, so what we're trying to do in selective breeding is we're trying to take the traits of an organism that we desire and cross those with another organism that has the same traits, and hopefully we'll get the exact same characteristics or characteristics that we desire to get from each. Now, this can come from 2 basic ways. It can come from inbreeding, which is the crossing of 2 individuals with similar characteristics. Okay, this can lead to increased probability of genetic disorders. We see this in human beings, what we're talking about with inbreeding, that's incest. That's when you actually have, the issue is that when you have relatives that are bearing children together, there is a higher risk of having the same allele or genotype issues. So, for example, if my family has a prevalence of, let's say, the gene for hemophilia, and then have inbreeding, right, or having relations with family members, there's a higher probability that they're going to have that disorder as well, and thus a higher probability that children in that situation would also have mutations or disorders from that. But inbreeding can be done in a way in some organisms where you're not going to have issues or disorders. You can also do what's called hybridization. This is where you cross 2 genetically different individuals to get a new pair. Now, inbreeding would be, you can do that with plants, things like that. So you could actually take 2 plants that have a particular trait that you desire, maybe stem length or a certain color of petal, and you cross those, you put those together in hopes that they'll just continue to have that same similar circumstance. Or in the book, it used the example of turkeys. You can take a male and a female turkey that are both plump and grow really quickly, and you cross them with the hopes that their offspring would be plump or larger sized, right? Because you're going to have them for turkey dinner, and that they would grow quickly. The problem with inbreeding, as we said, because there's a similarity in the alleles, there's a higher potential for inherited problems. We see this a lot in dogs. You can see this in different dog breeds. As we can see, hybridization really is, we've seen in dogs, you started at some point, okay, and this is an interesting terminology. I use this one because it used the gray wolf as a common ancestor. But what's interesting is all of these examples of breeds here are all dogs. We have no change from a dog, and then we get down here and we get a cat. Well, sorry, I think that's a pug. We don't see that. What's happened is we have these different breeds of dogs that have come from all around the earth. Okay, there are different types of wild dogs. And we've actually had breeds that have come down out of those dogs as you've done more and more breeding. And we now have literally hundreds of different dog breeds that have come because of hybridization. They've crossed various types of genetically different dogs, okay, that have different traits to get new traits from them. So that's selective breeding. And that has been around forever and ever and ever. Okay, one of the benefits of hybridization is in hybridization you may get an organism that has both traits. We get something in hybridization called hybrid vigor. The organism is actually stronger than its parents. Another way that sometimes modern genetics will actually apply this is they'll do something called cloning. You probably heard about cloning. Cloning is when an organism is produced that has exactly the same genes as the organisms from which it was produced. So their DNA of the parent organism is exactly the same as the DNA of the offspring. They did this first example down here in this drawing. We can see Dolly. And what they actually did is they would take the udder cell in a sheep, from a white-faced sheep, and then they removed the DNA from an unfertilized egg, so from a female sheep, and they actually fused the cells together with the donor DNA and then put it into an embryo. So they basically produced this embryo all in a petri dish, okay, basically, and then implanted that into a surrogate. So they implanted the cloned embryo into a surrogate sheep to go ahead and have the baby, and what they ended up getting was a clone of the white-faced sheep. This cloned sheep, Dolly, was a carbon copy, DNA equal to the sheep that they took the information from originally. Now, interestingly enough, what's interesting about this process is Dolly didn't live very long. Cloning obviously has some ethical problems. Yes, we have the technology to go ahead and extract DNA and cut DNA apart and put DNA back in, and that gives us a lot of ability to do some things. But there's also some ethical, like what are we doing here as we are actually playing God and creating organisms? So obviously cloning is something that is it's pretty early on in as far as the technology. We're seeing cloning in animals and things like that, but there's also cloning obviously happening at the same point here. So we can see this with humans where they can actually do that, and that's got some serious risks and things we need to talk about as we progress. The third thing way that genetics can be applied in determining traits is what's called genetic engineering. Genetic engineering is where they actually take the genes from one organism and are transferred those genes onto the DNA of another organism. And you can see this in bacteria. This is an example here in bacteria where they'll actually take, and you don't need to know this whole process, but what they actually do is they'll take the DNA out, a section of genes that they want. They'll remove that section, okay? So you'll see right here, they cut this section out. Then they actually are going to go ahead and graft that section back on to another example of DNA in this particular bacteria. And then it's reinserted, and the bacterium will go to this regular process of development that it normally does. And it now actually has, represented by this little black, a foreign gene, a gene that is not actually natural to that organism, and you actually get a plant out of this process that's made and expresses the gene and the traits that are desired by that particular process. So genetic engineering has happened in bacteria, and they've used this. One of the biggest things they've done is they've used this to actually produce insulin and large amounts of insulin, which is great for diabetics. So that's a positive way of genetic engineering. They've also used this in other organisms. They've used this to develop blood clotting proteins for people with hemophilia, which I think is a good thing. And they've actually put these types of inserted genetically altered or mutated organisms into plants like tomatoes and rice to develop particular cold hardiness or the ability to ripen fruit quicker, things like that. So they do have genetic crosses that are happening in other organisms. Now, someday, okay, there's ultimately, and this is not far off, they want to use this in gene therapy. This would be the ability to actually use genetic engineering to correct disorders in humans. So they could actually go in, they already do this where they do actually a karyotype, or not a karyotype, a gene fingerprint of a woman, of their child, and then they can actually determine the diseases that are in that child if the child has genetic disease, and then they would go in and actually make changes to prevent that disease. Again, there's some serious ethical concerns about this. What does this look like? What is this going to do? First of all, are we playing God in this? How far do we go? How far does this lead before it becomes we're stepping into things we shouldn't do? When does gene therapy become, hey, I want a really tall kid that can play basketball, and so I can go and order a particular child or a certain height or a certain intellect or intelligence, and then we're basically producing robotic children that we order. That's a stretch, but how far is it? So gene therapy is something that could have some benefit, but we have to be really wise in how we do this. Another thing that's just finished recently is the human genome, and they've really human genetic applications have been happening. They use DNA technology for a lot of good reasons. For example, DNA technology allows us to actually study the human genome and get in detail knowing, hey, what are the particular genes and traits and where are they carried on DNA? And all the DNA in one cell or organism, we call its genome. That's the entire organism's genetic fingerprint. They've also used this in actually what's called DNA fingerprinting. Okay, and I spelled it wrong. Let me fix that. Fingerprinting. This allows people to be identified by their DNA, and so we see this in court cases. We see this in identifying parents or children. They can actually use DNA. There's been court cases where someone's been accused of murder and he's been in prison for 15 years, and they come find new DNA evidence and realize, hey, it couldn't have been him. So there is a lot of other really interesting applications and advances that can happen with genetic engineering and gene therapy and understanding just human genetics. But we always got to be careful and cautious that we're wise in the way we apply technology, because if we do it the wrong way, it can actually be harmful. And so that is the end of this particular section, and also the end of this particular chapter as we've talked about human genetics. So please summarize and answer those questions, and I'll see you guys later.